Okay, welcome everybody. So we have posted homework 1 on the class webpage. I want to say a little bit about it. It has 3 questions, and this homework, as well as most of the homeworks we're going to give you, are going to be similar in that they're going to pose a question at the beginning. So there's 3 questions, and the question is pretty open-ended, but don't worry. Then we're going to give you a series of tasks that if you complete all of them, it'll guide you through answering the question. So at the very end, you will then be able to answer the original question. So the way to do the homework is to go straight to the tasks. You want to read the question so you can get an idea of the big picture. And we're trying to imitate how data analysis works in real life. You kind of ask a question, and then you go do certain things. You start getting an idea what the answer is. Then at the end, you can answer it. So what we're hoping that you'll do is you'll complete the task, and at the end, write a short answer to the original question. And you'll see what I mean by that when you look at the homeworks. But what you want to do is kind of look at the whole thing first before you start, so you can get an idea of what the entire process is like. All right, so that's the first thing I wanted to mention. The second thing I wanted to mention was that this Thursday, tomorrow, the next class, we're going to have a session where we're going to go over some of the key things you should know regarding the homework. So you should try it. So because if you have questions, you can ask it. It's not going to be a lab, but it's going to have a similar feel. We're going to go through, try to connect what Mark's going to teach you today and at the beginning of Thursday to what the homework is asking you to do. So did I forget anything? So here's Stephanie. You didn't meet her last week. She does exist. Did I forget anything? All right, Stephanie is our lead TA. TF. Okay, Mark. So today we have a guest lecturer. Mark, he's an expert in data visualization, and he'll be talking about, he's going to continue talking about data exploration, then some principles of visualization, which we want you to try to implement in the homework you turn in. We want your figures and your pictures to follow these principles as much as you can. And I'll let Mark start. Thank you.
Okay, thank you. Welcome, everyone. My name is Mark Streit. I'm going to teach the visualization part of the course, as Raphael said. And before I start, a few words about myself. So I work and live in Linz, which is in Austria. So Germany would be here, Italy here, and that's the small country in between. Vienna would be here, and Linz is this city here. And I'm an assistant professor there at Johannes Kepler University in Linz. And this year I'm staying as a visiting professor at SIAS in Hans-Peter Pister's group. That's the professor who did the data science course last year, but who is on sabbatical now. And I'm leading the visualization group there. We're doing mostly InfoVis research, and most of it is biological data visualization, so mostly cancer data. Okay, enough about me. I don't need to repeat this. Homework 0 is due today. Homework 1 is due next Thursday. So I think it would be good to start as soon as possible. All right, so I'm going to first talk about what are the goals of visualization. Then we move on to data types and data dimensionality. And the second part of the lecture today will be about statistical graph types. Okay, so what would be the main goals in visualization? And last time, Raphael already talked about Anscombe and the Anscombe's Quartet, which are these 4 data sets that he published. He was a statistician. And there's something special about these data sets. Who can remember what's so special about them? Anyone? Yeah? Yes, exactly. It's the same mean, same variance, same correlation, and same regression line. Yeah? That means if you throw statistics at it, then it will give you the same result. But if we look at simple plots, then you will immediately recognize that they are very different. So here we have one outlier. The other data is just gathering here. This is almost along the regression line, and we have an outlier here. And that would be this curve shape and so on. So it's completely different. But without plotting, you would have probably missed that. So this is one example that you find in every textbook, yeah, in the introduction section. Another example that I like very much is the normal distribution. And Raphael already talked about normally distributed data. And it's the Russian president elections from 2012. Yeah? And election data is also supposed to be normally distributed, usually. And right after the elections, there was a paper published which is just discussing the results. And there you see these quite odd peaks at round percentages. Yeah? So there were protests after that where protesters were just saying, we do not trust Shurov, who is the head of the election committee. We trust Gauss. It's a normally distributed data set. So of course, this is not a conclusive proof, but at least it raises some interesting questions, I guess. Let's put it like this. OK, what are the goals in visualization? First of all, it's presentation, and that's probably the most well-known to you. Yeah? You know facts about the data, and you try to communicate it. That's also what public media is doing. So Raphael introduced the New York Times graphics department, and they are doing really a great job in this presentation task. But we have also these other tasks which are exploration and confirmation. So in exploration, we don't have an hypothesis, and the task is to generate one using the visualization. And confirmation is when you have a given hypothesis, and your task is then to verify or falsify this hypothesis. So let's start with presentation. Yeah? And these are two examples, really early examples. So on the left-hand side, you see a drawing by Henry Gray from 1918. And the sole purpose of this graphic is, of course, to just explain how the heart is working and which parts you have. And this is from a very famous textbook in medicine. On the right side, you see a drawing of a female body by Da Vinci. And again, the sole purpose is to communicate something that Da Vinci already knew at that point. This is a really great example. Who has seen this before? OK, some of you, I would say 1/3 or so. Minard was Napoleon's map maker. And this is a visualization of Napoleon's march towards Moscow. Yeah? And what you see here, so it's just a static plot. What you see here, the thickness encodes the strength of the army. Yeah? And they started with 440,000 soldiers. So even for that time, that's really a lot of people. And then, of course, the army gets smaller and smaller, and only 10,000 of them returned. Yeah? And Minard wanted to actually try to present why this happened. And there you see the army split at some point in time, and some soldiers went back. Then we see that they approached Moscow, they entered Moscow, and almost the same number of soldiers went back. But then what's really interesting here is this temperature plot at the bottom, which is linked to the locations on their way back. So you see they had really low temperatures, and that, of course, again, reduced the number of people. And for instance, this is also really nice here. There's a river, and the temperature was pretty high at that time, so the temperature raised. It was pretty high, and there was no ice on the river, so they tried to build a bridge, and this went wrong somehow. So they again lost almost half of the rest of the army. Yeah? And this is just a story that you can tell with one single representation. Yeah? That's really nice. But let's move to newer examples. And that's the last one that I have for presentation. It's a heterogeneous heat map. We will learn about that next week. So what's behind this visualization is just a huge table. What you see here is a breast cancer data set. It's from a recent Nature paper from this TCGA, the Cancer Genome Atlas Consortium. It's a huge US consortium, and they're collecting data about cancer patients. And in this plot, they're just showing 4 different groups of patients. So they have about 500 patients having breast cancer, and they subdivided it in 4 different groups. And then for these different groups, they show different mutations on different genes. Yeah? So here you have the different genes, and you see this group of patients, they almost 50% in that group, they have a mutation on that particular gene. Yeah? And this is just a static image published in a paper, and its purpose is to communicate a point. So facts that they already knew. Very different to this is interactive exploration and confirmation. Yeah? This is this back and forth between data analysis, plotting, visualization, then changing the question, and so on and so forth. And yeah, we briefly talked about exploratory data analysis last week. So John Tukey said the greatest value of a picture is when it forces us to notice what we've never expected to see. And when we look at Wikipedia, you will find this definition. So EDA, exploratory data analysis, is an approach of analyzing data to summarize their main characteristics without using a statistical model or having formulated a prior hypothesis. So that's really to generate an hypothesis usually. Exploratory data analysis, it continues, was promoted by John Tukey to encourage statisticians to visually examine their data sets to formulate hypotheses that could be tested on new data sets. Yeah? And I guess the maxim here is detect the expected and discover the unexpected. Yeah? And this is also what you're supposed to do in your homeworks. And for exploratory data analysis, I've picked this example. So who has seen this before? Probably fewer people, yeah? Okay, so this is a map of the city of London, and at that time, it was in 1854, I guess, they had a cholera epidemic. Yeah? But they didn't know the source of the cholera. And they thought it's actually, they called it at that time bad air, which is pollution. So it was right after the Industrial Revolution, and they thought this was causing the problems. But John Snow, actually, he just collected the deceased individuals and the locations and plotted it on this map. Yeah? And there he immediately saw that there is a higher, or that there is a cluster around the water pump. Yeah? And this way they identified that actually the water caused the problems, or that the water was the cause of the cholera disease, and then they were also able to fix it, which is really, really nice. So there's Isaac Asimov, who used to be a professor at Boston University here, and he said the most exciting phrase to hear in science, the one that heralds new discoveries, is not eureka, but that's funny. And I guess this hits the nail right on the head. So this iterative process that you're supposed to learn in this class is, it starts with asking an interesting question. Yeah? You have a data set, and you're starting with formulating a question. Then you get the data, you scrape it from the internet, whatever, you need to clean it up, which is usually very tedious, and we will hear a lot more about that. And then you're just exploring the data. Try to understand what are the main patterns, what are anomalies, outliers, and so on and so forth by plotting it, visualizing it. Yeah? Then in the next step, you could build a model. You could fit the model, validate the model, and when you have found something interesting and confirmed it, then it's time to communicate the results. Yeah? But this is not just a step-by-step process, but of course you can move back to every step. For instance, if you see that the model doesn't work, then you could go back and change the data set. Yeah? Maybe there is something wrong with the data set, or even go back to asking a different question. Yeah? And I want to run you through an example. It's a historic example about antibiotics. And Wilbertin, he was a designer. And in 1951, he wanted to find out more about the efficacy of certain antibiotics. Yeah? So that was the data set that he had. On the left-hand side, you see bacteria. And here you see the different antibiotics, which is penicillin, streptomycin, and neomycin. And here you have the gram staining, yeah? Which can be positive or negative. And gram staining is not or is hardly used nowadays. Okay, so the gram staining just gives you how the antibiotics react, or how the bacteria reacts to the strain. Okay, so let's look at the data and the questions. So what are the data types that we have here? Any guesses? So what would be the first column? What's the data type in terms of programming? Yeah, it's a string, right? What's the data type that you find here? It's a number, it's a float, and what would be the gram stain? What? Yeah, it's a Boolean, right? Okay, so this is what you've told me. So what you see here is the minimum inhibitory concentration. Yeah? And the lower it is, the more effective the antibiotic is for the bacteria. Then we need to talk about the questions. Yeah? So what would be the questions that someone wants to ask? Yeah? Any ideas? So you have this data set. What would you like to know? Could you raise your hand? Anyone? Yes?
Is resistance to one kind of antibiotic correlated with resistance to the other kinds?
Yes. So how well the different antibiotics perform for the different bacteria. Yeah? And this is exactly the question that he wanted to get an answer to. And so how effective are the drugs? And he created this nice-looking visualization. And today we are not going to argue about if it's well-designed. Yeah? If you would say that it's not optimal to choose this radial layout, I would completely agree. But now it's just about the data. Yeah? What can you see here? So I will first explain it. So red would mean gram negative. Blue would mean gram positive. And here you see the different, how effective they are, basically. Yeah? Is there any trend that you can see here? Yeah? Yes, penicillin is better here, so you have large bars. And that's an interesting design choice here because he actually chose to small numbers to be a large bar. Yeah? Because it's small numbers are good because they're effective. Yeah? Anything else? What about streptomycin? Yeah, it's bad for both. Yeah? And on the right-hand side, neomycin performs better. Yeah? And that was the answer that he already had and that he gave in his paper that he published at that time. Okay, so on the left side, if bacteria is gram-positive, penicillin and neomycin are most effective, and on the right side, neomycin is most effective. Okay, and in 2009, people came back to the same data set, but presented it in a different way. They organized it like this. So they grouped the different bacteria by their resistance to P, S, and N. So that's the different kind of antibiotics that you have. So here we have bacteria that are resistant to all three. Here we have bacteria that are resistant to these two, and so on. And this is resistant to none of them. Yeah? Is there something odd that you see here? what's going on. If you take a second look at this data by using this visualization. Nothing that strikes you? Could you say it again? Which bacteria? Yeah, but the data is still the same, so. Anything else? Look at these two guys here. What's strange? So the organization of this plot is actually that they build these groups, the clusters. And as you see, they have a similar pattern. Yeah? So here you have a really large bar, and that's the same for all of them. Here they also have a quite similar pattern, also here. Yeah? But is there something strange going on? So the strange thing here is, and the question that you need to ask, is there something wrong with the data? Yeah? Because here we have, they have a resistance to streptomycin and neomycin, and these are streptococcus. Yeah? And this one is not. Yeah? So, and this one here is a streptococcus, yeah? But it's in the wrong group. So no bacteria here is a streptococcus. Yeah? And this was actually an error in the data, or an error in the classification of the bacteria. And they actually found this out 20 years later after Wilbert created his graphics. Yeah? And maybe if you would have done a better job in visualizing that, then you could have already figured this error out. Yeah? So it's not just about insights. You always need to look for errors too. Yeah? That's very important. But there is also a graphic. If we just do a scatter plot, and we will also learn about this later today, then you will immediately see this. Yeah? So there is a group. It clusters here. Here we have the neomycin concentration, and here the penicillin concentration, and we have these nice clusters. Yeah? And usually the different bacteria from the different group, they should react the same. Yeah? So, and that's something that's odd here. Okay, so far everything was static, right? But in exploratory data analysis, we're often dealing with this iterative process. Yeah? And it's called the visual analytics process. You start with the data, then you can change the data, you can clean it, and so on. And then you have two different branches. You have this visualization, so you can plot your data, and you can try to fit models, and so on. Yeah? And you can switch back and forth between visualization and statistics, basically. But at the end, it's always about the same goal. You want to gain some new knowledge, right? And in visualization, this is really critical, the user interaction. Yeah? So you need to interact with the data. So in many cases, it's just not enough to look at the static picture. That's why people created interactive visualization. And this is a really nice example from guys at the Pfister lab at Sears. And they looked at this task of comparing different genomes. If you compare different genomes, then you will have genes that are on one chromosome in one genome and on a different chromosome on another genome. And they actually they decided to go with this radial layout. So this is not just a simple plot, right? This is more sophisticated. That's a visualization that just answers a specific question. Yeah? And it was designed to work for this. So in my visualization lectures, today in the second part, we will talk about simple plots, statistical plots, and next week we will take a look at these more sophisticated techniques as well. So if I run the video, the analyst can really do an interactive analysis. So I will stop it again. On this outer ring, you have this reference genome, and on the inner ring, you have the second genome that you to which you want to compare the reference genome. And the user can select one chromosome, which will be inserted in this inner ring, and then you just see the connections to all of the other chromosomes of the other genome. Yeah? So you select a chromosome here, which will be inserted in the inner ring, and you can also filter your data to only show a certain section of the genome, and that's the way how you can try to understand what's going on between these two genomes. And that's, of course, more than just a simple plot. OK, let's talk about data types. And I briefly mentioned them in this example. And in 1996, Ben Schneiderman, who is a pioneer in the field of visualization, he came up with this classification. So he said we have 1D data, which would just be a list. And in a list, you can look at the distribution or whatever. Then you could have temporal data. If you have data that changes over time, for instance, you measure the temperature at a certain location, then this is temporal data. Then he said you could have 2D data. That's everything what you can project on a map. 3D data would be data that has a reference point in real world. An example would be if you go to a CT scan, you make a CT scan of your brain, and you take a voxel, which is a pixel in three-dimensional space in computer graphics, and you take a voxel in this visualization that you have, then this voxel actually corresponds to a certain point or certain location in your brain. So this is 3D data. Then we have ND data, which is what you would store in a relational database, just tables, which could be connected to each other if you have multiple of them. Then we could have trees, hierarchical data. That's what you have on your file system, where you have the root, and then it goes further down the hierarchy. Then we would have networks. An example would be your friends network on Facebook, and maybe others. And that was the data types that he proposed at that time. Later on last year, Tamara Munzner, who is a professor in Canada, she said that you have 3 different classes. You have tables, and tables would include 1D data, 2D data, 3D data, could also be a time series data set. Then we have networks, which would be your friends network, and text and logs. And that's a kind of data that gets more and more important with the web, of course. When we are talking about data types, there is this big difference between semantics and data types. So the data semantic is the real world meaning. It could be a company name. In the previous example, it was the name of the bacteria, whatever. But the data type in terms of programming, in terms of computer science, is just an interpretation in terms of scales of measurements. Is it a quantity? Is it a category? Is it Boolean? Is it a data structure? Whatever. So that's more on the engineering side. And there's this great paper by Stevens, who also used to be at Harvard University, and he talked about these different data types from a data perspective. So it's nominal, ordinal, interval, and ratio. And we will briefly go through them. So the data type could be nominal. An example would be apples, oranges, bananas, if we talk about fruit. And the only thing you can do with nominal data is you can say it's the same or it's not the same. Then we could have ordinal data. Ordinal data have a relationship, a smaller or greater relationship, like small, medium, large. You can sort this and it will make sense. This is also what you see here if we plot it. Then the third type is quantitative data. And quantitative data is different because you can do math on them. For instance, if you know the height of two persons, you could say this guy is 2-inch shorter than the other guy. Or you could say, I don't know, this desk is 2 times the length of my notebook. And so you can multiply it by 2 and it makes sense. This, of course, doesn't work with apples, oranges, bananas, bacteria, whatever. Okay, with quantitative data, you need to differentiate between interval and ratio data. Interval data has an arbitrary location of 0. An example would be January 19th, so a date, or a location, latitude, longitude. The 0 meridian through Greenwich, actually, that's just chosen by us. There's no other reason why this is the 0 meridian. And compared to that, you also have ratios. And with ratios, it's different because they are 0 fixed. That means their origin is meaningful. Like when you have a length, 0 length, that's a meaningful origin. 0 length is just 0. And that's the same for temperature and so on. There's an absolute 0 point and so on. Okay, so what kind of operations can we do with these data types? With nominal, as I said, you can only say if they're the same or not the same. With ordinal, we can also sort them, bring them into an order. With interval, quantitative data, we can calculate the distance. So if we have the location, then we can say, I don't know, Boston is 3 hours by car away from New York, or 4 hours. And if we have ratios, then we can also do proportions. Like I said, this is twice the size of my computer here. This only works with ratio, 0 fixed data. Let's do an example. So what data set would that be? Any ideas? It's not that hard to figure it out. So you have the age, you have fair, you have the class, first, second, and third. Yes, that's the famous Titanic data set. The data set comprises all patients that took the Titanic at that time. And when we look at it, it's nice to actually describe the data, or it's a nice example. So if we look at the first row here, that's the semantics that you usually have. Because if you just see 0 and 1, that's a data type. If you don't know how to interpret it, then it's for nothing. So we need to know about the semantics. The semantics would be sex, age, fair, class, and so on. Then if we talk about rows in such a tabular data set, then this is an item. Then the column would be an attribute, or a feature of this data set. Could be any tabular data set, of course. And now we will just try to figure out what's quantitative, what's nominal, and what's ordinal. So what would be survived? Nominal, yeah. What would be the class? That's ordinal, because first class is probably better than second and third class. Yeah. What would be sex? Nominal, yeah. You can't do math with it. You also can't sort it, so females are not better than males and the other way around. What would be fair? Quantitative, right. What would be class? We already had that. And there are some, like the town they went on the ship and so on. Okay, so I've done that for you too. Okay, so nominal and ordinal are dimensions. They describe the data, and we call them independent variables. And quantitative data are measures. That's the numbers that you need to analyze. And they're dependent variables. They always depend on a different variable. For instance, the fair depends on the location where you have bought it, and so on. Okay, when we talk about data, there's this difference between data and conceptual model. Yeah? The data model is what we've talked before. It's the low-level description of a data set. And the conceptual model includes the semantics and also reasoning. Yeah? So for instance, if we have a data set and it's one-dimensional float, and you don't know that, then there's nothing you can do. But if you know that this is a temperature, you can also add the meaning. For instance, you know that a certain value is very hot. And the same is for 3D vectors. And if you know that this is a space, so a location in three-dimensional world, then you know how to interpret it. Let's do an example. So how can you get from the data model to the data type in the end? So if you have the data model, which is just the raw data, and you apply a conceptual model onto it. Like say, my data is the temperature. And in the end, you could convert this to a data type. So you could say it's a continuous to 4 significant, so it's a continuous number with 4 significant figures. Yeah? Or you could say, I just convert it to hot, warm, and cold. Yeah? So I can move, or I can transform this raw data that I have here to an ordinal data range. But I need a conceptual model for it. Or I could also convert it to a nominal data, burned versus not burned. Okay, data dimensions. So if we take this Titanic data set and we just take one column out of it, then we have a univariate data set. And with a univariate data set, we can plot it as a histogram. So we have the numbers of patients here, and we have the age here, and it's supposed to be normally distributed. Then if we have bivariate data, so if we take a second dimension out of this data set, the first one was age. If we also take the fair, then we could create a scatterplot, right? If we take a third dimension, then, well, what could you do? You could create a 3D scatterplot. But I advise you to never use a 3D scatterplot, and there are really good reasons for it. And we will discuss that next week when we talk about effectiveness of visualization. There is a better alternative. So what would be a better alternative to encode these 3 data dimensions? Yeah?
Double axis plot.
A what?
Double axis?
Yes, what would be a parallel coordinates plot, yeah? Where you have 3 different axes, yeah? That would be an option. We will introduce that next week, so parallel coordinates. But what else? A very simple thing. Yes, exactly. So we take the scatter plot and we just encode the third variable or the third dimension using a different visual attribute. And we call it visual attribute. We're going to talk about them also next week. So you could use color, you could use shape, you could use size. You could, of course, also mix them. So you could create a scatter plot where you encode like 7 different dimensions. Yeah, that's of course possible, but it's not effective anymore. And we will see examples next week. Okay, what would we do if we have 4 dimensions? Yeah, then it gets difficult. And there is a very nice principle. It's called small multiples. Yeah? You create a tiny visualization for every attribute that you have, or for every dimension. Yeah? And this is called small multiples. If you're interested in the correlation, you could also do a scatterplot matrix. Yeah? And we're also going to discuss this next week. Yeah? Here you can see the correlation between any of the variables. Okay, but if we have tables with hundreds of different dimensions, yeah, then at some point, of course, we can't encode it, and it doesn't make sense to encode it in one single visualization. And there are different measures to take, how you can solve this. You could, for instance, filter your data. Yeah? That's of course reasonable. You just take the columns or the attributes that are important for you. Yeah? And this, of course, depends on the question that you ask. Yeah? If you collect a huge data set, then of course not all columns will be relevant for you. And the other really powerful thing to do is aggregation. Yeah? Aggregation is, Raphael talked about it last time, so a way to aggregate the distribution would be mean and standard deviation. Yeah? If you have a network, let's say with all your friends, yeah, and it's a huge graph, then a way to aggregate would be to just group them by the city where they live, yeah, and to collapse them to just one single node for Boston, for New York, and so on. And this is called aggregation. Of course, you're losing details and you make it coarser, but at some point with big data, there's no other way around it. Yeah? And we will discuss this on next Monday in more detail. We will also talk about dimensionality reduction, multidimensional scaling, and Raphael is going to introduce that. Okay, now to the second part for today, which is statistical graph types. Yeah? And I'm not sure if we want that you end up like this guy during the course here. And these are, by the way, XKCD comics. So who is aware of XKCD? It's a science comic series. Yeah, if you're not, take a look at it. It's really worth to check it out. And even better, there's a matplotlib mode. Okay, it's a style where you can XKCD-ify any plot that you create. Yeah, and that's really great. That's awesome. So here we have Anscombe's Quartet. Yeah, and there's just one call that you need to add, and you get an XKCD version out of it. Yeah, which just adds this, I would say, uncertainty by this, by changing the line styles. That's really cool. But you shouldn't submit it for your homework, I would say. Okay, what different types of charts do we have? And of course, it's again a nice way to make categories out of it by looking at the question that you want to solve. And here the question would be, do we want to compare something? Do we want to look at the distribution? Do we want to look at the composition? Or do we want to look at the relationship? And these are, of course, general questions. Yeah? And when you take a real data set, usually you don't start with a general question. Yeah? You start with a domain-dependent question, I would say. Yeah? If you ask, I don't know, a biologist, what do you want to know from the data? He would say, I want to know if bacteria A performs better than bacteria B, and so on. Yeah? And then you need to transform this domain-dependent question to a domain-independent general question where you then can try to figure out what plot or what visualization you create. Yeah? And unfortunately, there is no automated way to do that. Yeah? There is no list of domain-dependent question, domain-independent question, and because then we would just have systems that can create the perfect visualization to answer our question. Yeah? And that's unfortunately not possible and will probably not be possible anytime soon. Okay, let's start with comparisons. What would be a good way to compare data? Yeah, let's say we have quantitative data and we want to compare it. Suggestions? Yes. So the simplest thing to do would be create a bar graph. Yeah? On the left side, you see this XKCD version. On the right side, you see one created with matplotlib. And I've always provided the link here where you can find the code to generate that. Okay, and the reason for this, why we are good at this, is because we are our visual system is very effective to interpret position and length. Yeah? We are much better in doing that than to interpret different shades of a color, yeah? Or different shades of gray. Then direction is also important. So in theory, there is no better direction for a plot. Yeah? Usually it doesn't matter. But the rule of thumb is if you have long labels, yeah, that's a very practical reason, then go for a horizontal plot because you can put here nicely the numbers and also other long labels that you have here. Yeah, that would be much more difficult and space consuming if you would rotate it, yeah? Because we can only read text in a horizontal manner. Yeah? It's a very practical reason. Then trends over time. Yeah? There's also this XKCD thing that I like very much. So, and in this course, you will also try to extrapolate data. Yeah? You will start with a data set and try to find a trend and maybe also predict something. Yeah? That could be a task for your final project. What would be the right plot for doing that? Yeah, if you look at time-dependent data, so data over time, yeah? Yes, but we're still looking at 1D data. Yeah? Yeah, it's a simple line chart, line plot, line graph, whatever. Yeah? So they all have the same, that on the x-axis we have this continuous axis, yeah? And that's different to what we have in a bar chart. Yeah? Here we have, as time is continuous, we have time on the x-axis. Also, mathematical functions are continuous. Yeah? So if we have a sine function, then of course this is also the best way to plot it is to go for a line chart. Yeah? And here would be, if you have measured data, of course you can try to fit the actual mathematical function behind it. Okay, let's talk about linear versus logarithmic scale. Yeah, if we look at this plot here, this would be linear scale and log scale, and that's just the Apple stock price from this morning or yesterday evening. Yeah? Maybe the price, as usually, dropped during the Apple event today, but this is not included yet. Okay, so what's the difference with linear and log scale? What would you say is better here? Is there a version that is better? Yes?
The log scale is more useful because you want to see how percent changes work within the stock price, whereas the absolute change is going to be different depending on how it was originally arised from $5 to $10 is much more in the stock price than from 555 to 560.
Exactly. So, but this very much depends on the question you're asking. Yeah? If you say, I'm interested in the overall changes over the last years, yeah, then probably this is more intuitive to read, yeah, the linear scale, yeah, because you see absolute numbers. But if you're interested in what are the increases per year, then this is much more effective because especially for the years before 2000, in that plot almost nothing is happening because it's way too low to actually see it. Yeah? And this is different if you have a log scale, then smaller numbers, so trends in smaller numbers are still preserved, but you will also see higher numbers here. Yeah? And that's log scale. Yeah? That would be the XKCD version. They have a nice explanation. So the science tip is log scales are for quitters who can't find enough paper to make their plot properly. Yeah? It's very nice. But still, the problem that you have if you pick the wrong scale is that you have very low numbers that you hardly can see, and you have a really large bar where you have problems to fit it. And if you fit it, all the other smaller numbers will disappear. Yeah? Okay, bars versus lines. So you will see this is really simple stuff, yeah? But this is very important when you think about that. So lines imply connections. Yeah? It's called Gestalt principles. Yeah? And if something is connected, this is very strong for us. Yeah? And if you connect categorical data, like if you connect females and males here, we will immediately introduce data that is actually not present here, yeah? Because it's not continuous. As I said, a line chart is only valid if you have a continuous axis. Yeah? But if you have categorical data, then don't use a line chart. Don't connect your apples, bananas, oranges, or whatever with lines. Yeah? Just use bars. And the same is true for females and males. Okay, here is another example which is bad. So that's the average ratings for the Wheel of Time. Yeah, who has read the Wheel of Time? Okay, okay, it's not that popular, I guess. I haven't read it, so I don't know if it's worth to read it. But you will see that at the beginning it was the average rating was quite high, and here it dropped, and then it again was quite high. Yeah? But again, there is no reason to actually connect it because the volumes, it's not a continuum. Yeah? You could sort it, but still, if the volumes have not appeared at, I don't know, once a year, and I'm sure that was not the case, then that's not a continuous axis. Yeah? So there is no reason to actually connect these different volumes that you have. All right, aspect ratios. That's also very interesting. So here you see the same data, the same plot, but with different aspect ratios. What would be the best version? Any guesses? And this is a choice that you can do by your own when you create your plots with matplotlib. I mean, there is a default, but of course the default does not need to be the best. So what is the aspect ratio that you should go for? Any ideas? What do you think? What do you like best? Yeah, let's start like this. This one? Okay. Other opinions? This one, this one? Okay, so it's hard to tell. But there's a very simple rule. It's not an absolute rule. It was published in a study by Jeff Heer and Agrawala, and they actually proposed to go to optimize it to 45 degrees. Yeah? You should, it's called banking to 45 degrees. So two line segments are maximally discriminable when the average absolute angle is 45. So this is too flat, this is too steep, and this would be perfect for us to judge the different changes in the angles. Yeah? And that would be an example here. That would be an example. Cleveland proposed that, of course. That would be an example here where they show that this is more error prone than this. And they run a user study where you just were supposed to interpret these changes in the curve to figure out the numbers behind this plot. And this would be the optimal aspect ratio where you have the curve maximized to 45 degrees. Yeah? And you should take it with a grain of salt because it's not an absolute rule. It's not a no-go if you don't do it. But this is just the way people suggest to do it. Yeah, other don'ts, and we will talk about this next time. We will spend some time on discussing choices of color. Yeah, but the most important and most simple rule is don't add clutter to your data set that is not just the data and that is not needed to convey the data. Yeah? And there are really nice examples, and we will do that on Thursday. There's, for instance, a banana chart that has a background of bananas and things like that, and this is called chart chunk. Okay, so correlations. What would be the best way to look at correlations? Yes? Okay. So you just tried, to choose the aspect ratio so that most of your curve will be along a 45-degree angle. Of course, that's not always possible, but on average, this should be the rule. And if you do that, then it will be easiest for your users, for the guys who read your plot to interpret the actual data that is behind the graph. Yes. Yeah. Okay. So correlations. What would be a good choice for correlations? Yes, Scatterplot. On the left side, there's again this nice version of XKCD. Yeah. So he he actually plots fruits according to if they're tasty and if they're difficult or easy to open or to peel. Yeah? So for instance, the pineapple, it's very difficult to eat. Then you would have grapes. Yeah? If you have seedless grapes, then that's optimal. You just need to put it in your mouth. And this is of course highly subjective. Yeah? If it's tasty or untasty. Okay, and on the right-hand side, you again can see the data plotted with matplotlib for the Titanic data set that we discussed before. So usually with scatterplots, you have the problem that it gets crowded. Yeah? And this is called overplotting. Yeah? If you have lots of points, then you could decrease the size of the points, of course, but at some point they will disappear and it will still be crowded. Yeah? If you have a couple of millions of dots, then it's just black. Yeah? Yes. And there's also another reason for it. Yeah. Okay, so if we change it, so we could make, if we use color, then we could add this black border around it. Yeah? But this again just clutters, and there is another option to use this light gray border, which makes it slightly better, but I'm also not a fan of this. Yeah? There is a better way. What would be a better way? Any ideas? To fix this problem of overplotting? Yes? Yes. That's the correct answer. So overplotting is when you just get black and it uses Raphael's printer, then we could decrease the alpha value, which is the transparency. That's what you said. And then we can see patterns in the data that we haven't seen before. Because here, even some outliers, if you have a very huge data set and you have a part of your population that is actually an outlier, you can't see it because it's just cluttered. Yeah? But here you see you have these, it's the price of gold and carat plotted. Then you will see that it's actually, it will form these patterns here. Then you can also add trend lines, and I guess we will talk about more about this later in the course, in the next month or so. And again, don't use 3D plots. Yeah? The problem is, I can summarize it here, the problem is, of course, occlusion. Yeah? Without rotating this plot, there is actually a good chance that some parts of the plot are hidden by other parts which are plotted closer to the viewer. Yeah? And you also have perspective distortion. That means things are getting smaller if they're further in the back, yeah? Because it's a perspective thing, perspective graphics that you have here. Yeah? And for us, it's actually hard to tell what's the value of this guy here is, because we are not really good in touching this. Yeah? But more on that on Thursday. Okay, the last one is composition. Any idea? Or no, we have two. We have distributions and compositions. Compositions, what would you prefer? Yeah? Yes, but the downside is that you actually need interaction. Yeah? So, I've tried that. So we did some tests with scatterplots and biologists, and it was very hard for them to actually use the interactive features to rotate it and then again orient yourself in the data. Yeah? Because the patterns look completely different and you have other parts hidden. Yeah? So it's not really a good solution. Yes? Projecting the plots onto the surfaces, like what you have here? Is that what you mean? Or? Yes, that's what people sometimes do. Basically, what you have here would be a projection of the 2D scatter plot between these two variables, right? But then why not just showing 3 different scatter plots showing you the 3 different correlations between the variables. Yeah? That's probably more effective. But we will have examples next week, or on Thursday already. Okay, compositions. What's the type you would prefer for compositions? So compositions mean you have something that is whole, and you need to split it up in different sections or different parts. What would be the thing that everyone is using? Pie chart, yes, correct. So again, as always, I've picked a really nice example. So that's the pie chart from XKCD. What would you say, is Donatello a Renaissance artist or a Ninja Turtle? Yeah, and that's actually the distribution of people who think that, or whose first association would be a Ninja Turtle, yeah, which is really interesting. And I guess this is also dependent on the age, I would say. Yeah.
You also need another sliver for CS109.
Yes, true. Okay, and on the right side, you see this quite boring passenger class on the Titanic. Yeah. Okay, the question is, can we do better? Yeah, and actually we can. In, I would say, 99% of the cases, it's better to use a bar graph. Yeah? And there are some people in my community which are really religious about that. Yeah, they say never ever use a pie chart. Yeah? And but there are rare cases, I would say, where this makes sense. One case is, for instance, if you're interested, if company B and C, how they sum up together and what's their share of the whole, yeah, then it actually would make sense. But in any other case, it's much easier for us to interpret bars because length is a very effective visual variable, and angle is not, because here we need to judge angles. Yeah? And we need to transform an angle into a number. Yeah? And we are not very good at this. Okay, there is a thing that is even worse than bar charts, and it's a donut chart. Yeah? Don't do donut charts. Yeah? What would be the reason why this is worse? So what makes the donut the donut? It's the hole. Yeah? And by removing this inner part, we're actually not really able to judge angles anymore, but we need to judge areas. And we're even worse at judging areas than angles. Yeah? And next time, we will see that there is really a ranking of these visual variables, that we are best at judging position and length, and then angle, and then areas, and at some point there is color. Yeah? And color is usually something where we're really bad at. And people overuse colors, usually. Okay, then you have something like a stacked bar chart. Yeah? If we have a proportion of a whole, yeah? Like here this is 100%, then this would be 60%, 40%. Yeah? And that's very intuitive for us to read. Yeah? And if you have a stacked bar chart, so that's like just putting them on top of each other, and we also did that, or Verena did that last week with the university ranking, then we have, it's easy for us to compare the red part. So who died on the Titanic, yeah? First class, second class, third class, because we can just compare the length of the bars. But it's harder for us to actually do that for survived people, yeah? Because you don't have the same zero base. You don't have the same baseline, and that makes it hard. Okay, there's this stacked bar chart versus pie chart, and I said that the right one is almost in any case, almost in every case, more effective than the bar chart. Yeah? So if you have the choice, go for the bar chart, for the stacked bar chart. Okay, that's a comparison of which bar chart you should actually use for the different kinds of tasks. Yeah? And that's very interesting, I think, because if we have different categories, yeah, and different items, so it's just a table, yeah? And if our task is, like in the university use case, in the university ranking, we had this case, so the items would be the universities, yeah, Harvard, Yale, and so on. And these categories would be the different measures that you have. It would be like citation count, faculty-student ratio, and so on. Yeah? And if we stack them up, then the longer the bar, the longer the total bar, the better the university. Yeah? That's very intuitive for this task. Yeah? But if we want to compare the different universities for just one attribute, then layered bar charts are better. And then we switch to this small multiple approach, where we have for every category, we have a different plot. And here we have the same baseline, and that helps us to actually see that here there is not much more variation going on for the green category, but it's very hard to judge if it's stacked. And the group bar chart, it helps you to compare the different, all the different attributes for one university, but it makes it hard for you to compare this last category for the different universities. So what I want to say here is that the choice of stacked bar chart, layered bar chart, group bar chart, it very much depends on the question you're asking. And this is really important. Okay, yeah, I wanted to show you a little video. So together with Hans-Peter Pfister and his group, or my PhD student, thought about how can we improve this ranking. And this is actually to work with rankings, to understand them. And this is actually the same data set that you have imported in Matplotlib last time together with Verena. It's this QS World University Ranking. And, whoops. I'm not supposed to click on this one. Sorry. Here we go. So this is the accompanying video to the paper. And I will just fast forward. So you start, and this is what I mean by interaction, and you will immediately see what I mean. So we start in this prototype with all the universities. They are sorted by alphabet here. We have a column with all the different countries where the university is from, and then we have the different attributes, like employer reputation and so on, faculty-student ratio, citations per faculty, and so on. Then you can just throw it away, you can insert it, you can rearrange it, and this is much more convenient than working with a static plot, of course. Then you can also try to handle that you have thousands of different universities by switching to this fisheye. We could say, I want to filter it down to contain only universities from the US. And that's what the user is doing here. So the list is immediately reduced. You don't need to go back to your Python or whatever. This is, of course, an implemented prototype that makes it easier. Then here you can just say, sort it according to one attribute in your data. And it will create you a new ranking that is just considering this attribute. But now the user wants to create or wants to reproduce this QS World University ranking, and you just say, I want to make a new stacked plot, and I want to drag every attribute that I have to this stacked plot. And when you double click on it, it will just resort it, and then you need to type in the weights. So as Verena told you, this World University rankings, it puts weights to the different attributes that you have. And this would be the final result of the ranking. So MIT is first, Harvard second, Yale University. So that could be an error in the data, I guess. So the nice thing here is that when you actually drag this little guy here, it changes the weight. And it immediately updates the ranking. You could also say, I want to make a snapshot of the ranking, so I want to remember the ranking, how it is. And then if you're a student looking for a university, you just change parameters. You could change some weights. If you say, well, for me, it's not interesting if they publish anything. I don't care. Then you just get rid of this attribute, and you will see with this what we call slope graph how the ranking changes if you make certain changes to the attributes or to the weights. Yeah? And this is what we call interactive visual data analysis. Yeah? And this would be, I would say, the next step. After you do static plots, at some point, you would like to go to interactive data analysis, but this would go beyond the course, and you could learn about that in Hans-Peter Pfister's visualization course, which he's teaching next summer. Okay, let's go back to the presentation. If you have quantitative data that changes over time, a good choice is a stacked area chart. Yeah? What you see here is a data set changing over time. In that case, you have three different parameters. For instance, if it's I don't know, the money the government spends on universities, the money the government spends on defense, and so on. And if you stack it, you see the overall money that the government spends. Yeah? But you also see the shares between the different categories. And this is called a stacked area chart. And unfortunately, you need quite some code to achieve that in matplotlib, because it's not a standard plot there. Yeah? But if you click on the link here, there you will find the code that actually creates this stacked area chart. You could also do that 100%. So the difference is that you always fill it up to 100%. This is called space filling. And this is then the 100% stacked area chart. If we take a look at the stacked area versus the line graph, then you will see what the advantage and what the disadvantage is. Yeah? So try to give me the numbers. So how the green share evolves over time. Yeah? So if you would create a line graph, how would the green data look like? How would the curve look like? Yeah? Yes, it would be flat. How would the orange curve look like? Yes. But it takes you some mental effort to do that, right? So the orange curve would be decreasing here, but you have this increasing trend here. And depending on the question you ask, sometimes it's easier if you want to look at market shares, then this one is more effective. If you want to look at the overall trends and compare them among the different categories or among the different data that you have, then a line graph might be more effective. So this, the important take-home message is that it always depends on the question. And this is another example where you can see that this is actually much more effective to see what the absolute trend in your data is, and this is probably the hardest to read. Yeah? So the stacked version is better to read than the individual bars that you have here. Okay, the last one would be distributions. I already showed it. What's the best way to show a distribution? It's a histogram. Yes. And we had this example with you entered your height, and Raphael showed you the histogram. Here we have passengers versus age. What's the variable that you can change with a histogram? Yes. If you go for 20 bins, then it will look different. So what you do is you split up you assign your data to more bins. Yeah, compared to here. Here you have 10, here you have 20. But the interesting thing is that the trend can look very different. Yeah? So it's important for you sometimes to choose the proper number of bins. Yeah? Because here you will see that actually this is increasing for infants, I would say. But then you have below 10, it remains the same. But you completely lose this one here if you have chosen the wrong bin size. Yeah? You completely forget this guy, or you can't see it anymore. Okay, so what happens if we increase the bin size? If we have the data, we could increase it to finally end up to a continuous density plot. Yeah? So a histogram with a really large number of bins, it's at some point it's just a density plot. Yeah? But this depends on the resolution of the data. If you only have like 100 different people entering, or 200, I think we had 250 people entering the height, then of course you don't have enough data to actually get a perfectly fine density plot. Yeah? You would need to interpolate it, and when you interpolate it to make a smooth curve, you're actually introducing data that is not there in your data set, which is not good. Okay, if you have a 2D distribution and you're interested in the, so if you have a 2D correlation and you're interested in the distribution, you could create a 2D density plot. Yeah? The idea is just that you have a grid of bins and you put these little guys here in the bins, and then you assign a color scale to actually make it darker if it's more dense here. Yeah? And that's a very nice way to actually also deal with the overplotting, yeah? Because we can't see what's going on in here, it's just all black, but here we see that there is actually a nice center with a very high density. Another option would be to just show the correlation and then go for scatter plots here. And that's also an option in matplotlib. Yeah? You just need to turn on a flag and you will get these histograms for both of the dimensions of the scatter plot. Okay, the last thing we're going to discuss for today are box plots. Very important. It's also called box and whisker plot. And again, I've picked a really nice XKCD, so it's amazing that you really find for every graph type you find an XKCD example, which is really cool. Okay, here you would see a box plot showing the different, now everybody should have read it already. Here you see a box plot showing the different distributions, so a uniform distribution, normal distribution, Poisson, and so on. Yeah? Okay, I will walk you through how you are supposed to read the box plot. Yeah? In the box plot, so last time we had this height data set, and Raphael told you that you can summarize it by two numbers, which is the mean and the standard deviation. Yeah? But here in the box plot, we actually take the median. Okay? Who can explain what the median is? So the mean would be the average and the median, yeah? Yes, where you have 50% of your samples. So it would be if you sort your data set, so if you sort your data set, if you have this height data set and you sort it by height, then you take the one in the center and this is the median. Yeah? So this is the line that you have here. And then you have quartiles. Yeah? And if you look at the corresponding distribution here, then this is 50% of your data. It's quartile 1 to quartile 3, and this is called the interquartile range. Yeah? And this is the actual box that you have here. Yeah? Then we have the whiskers. The whiskers actually, they go to Q1 minus 1.5 multiplied with the interquartile range. And this is actually 99.3% of your data if you have a normal distribution. So this is really then, the rest is just outliers. Yeah? And this is much better to summarize your data if you have, for instance, a skewed distribution, if you have, I don't know, lots of outliers, then because outliers are not influencing the quartiles. Yeah? But it's different with the standard deviation because the standard deviation is actually influenced by the outliers that you have. Yeah? So that's much more effective to actually plot the distribution. And there is a comparison. What people sometimes like to do is this version, yeah, which is actually a bad choice. Here we have bars, and we add the standard deviation. So that would be the average, yeah? And then we have the standard deviation. for the different example distributions here. So that's a uniform distribution. If we look at this plot, that's exponential, Poisson, and that's the normal distribution that we had. So if we would just show it as a bar chart, then this is actually a bad idea. Has anyone a guess why this is bad? Why we shouldn't do that? Okay, I think it's not so easy to tell if you hear it the first time. The reason is because bars always start at 0. Yeah? And there could be, or in many cases, there might not be a single sample in your data set that is actually somewhere between here and here. Yeah? So you're showing a bar where there is actually no data. Yeah? And this is just wrong. You shouldn't do that. You shouldn't use bar charts with standard deviations for showing distributions. It's much better to actually show box plots. Here we have the median, and here we have the left and the right side of the box, Q1 to Q3, and then we have the whiskers. Yeah? And there are actually different ways to draw the whiskers. Yeah? There are some people who draw the whiskers to the minimum and to the maximum. Yeah? But usually it's better to use this way that I've presented here. So Q1 minus 1.5 multiplied by IQR, so this 99.3%. And the reason for this is because you can show the outliers then with little dots. Yeah? And it tells you more about your data set. Yeah? And it's more effective to actually convey more information in this plot. But in your homework, you can actually choose what you want, but just make sure that you document it. Tell us what the whiskers are. Yeah? Otherwise, we will not know because there are different interpretations of that, which are both valid. Yeah? But I would say, so I prefer this one with the outliers dotted. And there is just a switch in Matplotlib to make that happen. Yeah? OK, that's the last slide that I have before I'm going to summarize. It's the violin plot. And the violin plot, it's just a box plot plus the probability density function. Yeah? So here would be the box plot, but you could also add the density function here and then really convey more information. Of course, it's not so useful if you have a normal distribution, but if you see that there are these peaks here, then it might be an interesting choice to actually do that if it's a different distribution in your data. OK, so on Thursday, we're going to talk about effective versus non-effective visualizations. Yeah? We will have nice examples from the media which are good and which are bad. Then we talk about these visual attributes. That's the language, the alphabet that we have for creating visualizations. Then we will talk about color, and in the end, about design principles. And that's the banana plot that I mentioned. OK, then, see you on Thursday, I guess.